Our next practical is about earthworm dissection. Before dissecting earthworm, we have to learn a little bit about the earthworm classification, its availability, habitat, morphology, and anatomy. The earthworm classification. Earthworms are animals, so they belong to kingdom Animalia, and their phylum is Annelida. Annelids are the animals that have annuli on their body. Annuli are ring-like structure. A singular is annulus. So their body is divided into ring-like structure. There are septa that are running from exterior to interior, mean these bodies are completely from outer side and inner side they are they are separated into segments. So due to these annuli, these animal they are classified in phylum Annelida. So they are ring-like structures. Animal that have ring-like structure would be classified in phylum Annelida. The class is Clitellata. Clitellata are the members of this class. They have broad saddle-like clitellum on their body. For example, we can see in this earthworm specimen that at the center toward interior side there is a saddle-like, it is a broader area, and this area is actually glandular. It will secrete sticky secretions, and we'll see its function later on. So if clitellum is present on annelids, then they are classified in class Clitellata. And then order Opisthophora. Opisthophora mean the male genital pores, they are present behind this clitellum, so it is Opisthophora. And then is the family Lumbricidae, it is related to earthworm, the name of earthworm, and then genus Lumbricus, it is again earthworm, and then this is not subject, it is species. Species is Lumbricus terrestris. So Lumbricus mean earthworm, terrestris mean they are terrestrial earthworm. It means there could be other earthworm that are present in aquatic environment. So this earthworm that we will dissect is present in the soil, it is present in terrestrial area. So as the availability of the earthworm is concerned, it is present all over the world, and they prefer moist soils with rich organic matter, and they are that soil should not be dry and sandy, and they are mostly nocturnal. They would be active at night, so their collection is very easy. If we see at night, we would we can use a flashlight where the soil is rich with organic matter. We would find them during the nights and nights of springs and early summer. So and mostly when weather is warm and humid. And then during daytime they could be collected by putting a bucket of water on the rich soil, so their burrows would be flooded and they would come out. And then there could be different types of species present in same soil, so we have to observe whether it is Lumbricus terrestris or any other species, because it is important, because it can cause confusion while dissection. With their internal and external features, morphology and anatomy could be different from species to species. For example, number of seminal vesicles, spermatheca, they are different, and there is placement of different organs at different area. The external features, for example, genital pores, number of segment in their bodies, they could be different from species to species, so it can cause confusion if we dissect instead of Lumbricus terrestris any other type of earthworm. So we have to first confirm species. Then The common observation what we can do and can differentiate them from other type of worms, uh, it is that their body is segmented, having annuli on them, and they have setae on their ventral side. So if we put an earthworm on a sheet of paper and observe the mechanics of crawling, we could see the peristaltic movement of circular and longitudinal muscle, and these muscles they contract alternatively to help the animal to crawl, and the setae they anchor them, the different segments of their body, to help in the burrowing and in the crawling. So they could produce scratching noise on the paper. So when we have collected the earthworm, we have to give them anesthesia. So the earthworm they can be anesthetized by submerging them in 10% ethanol for 15 minutes, or we can use chloroform, but we don't have to touch them with the chloroform. We have to put chloroform on a cotton wool and place near to the earthworm in a closed container, and then their body could be washed with the water and they could be dissected. So chloroform and 10% ethanol they could serve as anesthesia for earthworms. Then we have to study their morphology. Discuss that they belong to phylum Annelida, that is the phylum of segmented worms. So these segments are present internally and externally, and these segments they are dividing body into segments through a central wall, that is the septum, or a central wall, that is dividing wall. These central walls are the dividing walls. They are not present only in the head and the tail region. The rest of the body is divided into complete segmentation. So on the head and tail we can see the segments, but they are only on exterior side, but at the rest of the body these segments, their septa or their separations are running inside also. So these are complete separate independent segments. So there could be other members of the phylum Annelida, like clam worms, tube worms. They are living in marine environment, and leeches are also members of phylum Annelida, and they are present in the fresh water. So for while observing the earthworm, we have to the morphology of earthworm, we need a dissecting microscope or a magnifying glass. So first we have to identify the dorsal side of the earthworm and ventral side of the earthworm. Dorsal side is bit rounder, and it is the uh, darker as compared to ventral side. Ventral side is flatter and it is lighter in color. And the dorsal side, on the dorsal side we can see a prominent blood vessel that is more dark than the even dorsal side, and it is called as dorsal blood vessel. There could be blood vessels present on the ventral sides, but ventral side is lighter as we know. And then the anterior side is more round as compared to posterior side. Posterior side is flattened, interior side is cylindrical, it is round, and then it is more pointed as compared to the posterior side. So we can identify the anterior and posterior side of the mouth and the anal side. We can identify them by their morphological characteristics. So on the anterior side there is the first segment that is called as peristomium. Stoma mean mouth, peri mean around. So peristomium is the segment that is present around the region of mouth, or it contains mouth inside. And then there is a lobe that is hanging from peristomium, it is called as prostomium. It is not a complete segment, it is just a lobe that is hanging from peristomium. So it is prostomium, mean it is before mouth. So this lobe is before mouth, so it is called as prostomium. The head of the earthworm is not very specialized, it is lacking specialized sense organ, and it is considered as degenerate head, and it is not a typical of the annelid, and we can find the segment at the posterior side, and it is the anus. The adult earthworms that are sexually mature, they have a distinct, very visible type of segment, and they are called as that is a swelling like region that is called as clitellum on the dorsal surface of the earthworm. The sexually immature or the young earthworms, they would not have this region that is clitellum. This clitellum is uh, of white or orange color, but it is lighter than the rest of the body, and it is located about 1/3 of the way down the earthworm, and generally it extends between the segments. If we come from anterior side, they come to the clitellum. It starts from 33 to 37 number segment in case of this earthworm, that is the uh, lumbricus tristis. As we said, this clitellum, it is excretory organ, so it produces mucus sheets that surround the worms during mating, and is responsible for making the cocoon within which fertilized eggs are deposited. So fertilized eggs are protected by that cocoon from dryness until they get hatch and produce new earthworms. So the purpose of this clitellum is producing the mucus for protecting, for making a structure cocoon for protecting eggs. Side up, we can see that the ventral side is a little lighter than the dorsal surface. If we run fingers on the ventral side of the body, especially on lateral sides, we can feel bristle-like projections. So these are the projection, they are called as setae, and these setae, they help prevent slipping of the earthworm and help in anchoring of earthworm while crawling. We can observe these setae. There would be a pair located on lateral side of the earthworm. For example, one pair is on left side and one pair is on right side of the earth. So on each segment there are 4 setae present. These setae would not be present on peristomium, the first segment, and the last segment, that is of anus. The rest of the segment, they contain 2 pairs of the setae on lateral side of the body, on the lateral side of ventral side of the body. Earthworm, we can observe reproductive openings. The earthworms, they are monoecious, mean they contain both male and female reproductive system. However, they cannot self fertilize, mean their sperms, they cannot fertilize their own eggs, so they have to get sperms from another individual and transfer their own sperm to another individual. Here we can see the openings of male uh, genitalia, So male genital pores, they are present on the 15 number segment. They are very visible, they are conspicuous, and they are present in this species on 15 number segment. At the segment number, so here the testes, they would release the sperms, and these sperms, while copulation, they are transferred to another individual, and when they are transferred, they would go to another organ of the earthworm, that is called as spermatheca in the second individual. So this animal, it is also having spermatheca pores. So here are the, at the segment number 9, at the it is segment number 9, and then segment number 10, and on the segment number 11. So in these animal that are Lumbricus tristis, there are 3 pairs of spermatheca. Spermatheca are for storage of sperms of another individual that this earthworm got during copulation. So these pores, they are not very visible. So sperms from another individual while copulation, they are stored in these spermatheca, and later on they come out on the outer side of the body, on the body wall, and then they would reach to the oviducts, or the female genital opening. So on the segment number 14 in this species are the female genital pores. There are 2 female genital pores where the oviducts release egg, but these genital pores, they are not visible like the male genital pore that are very visible. They are, female genital pores are inconspicuous. So the sperms of another individual that this individual got during copulation would come toward, they would run, they would come out from the pores, spermatheca pore, and then they would run toward the oviduct or female genital pore where the eggs would be present. So it would fertilize them, eggs would come out, and then they would reach to the lateral side of the body. Here we can see, if we see carefully, there are the grooves, there are lines, depression in the body. They are called as seminal grooves. So the sperms and eggs, they could run in these seminal grooves toward clitellum. So they would stick here, and they would run toward clitellum inside these grooves. So they reach to the clitellum. So for copulation, the animal arrange their body such that the male organ of one individual, male genital pore of this individual, would get aligned to the spermatheca of another individual, so that the sperm from this individual could go to get stored in the spermatheca of another earthworm. So these sperms, they would later on fertilize the eggs of this uh, earthworm, and then the sperms of this individual, they were stored in the spermatheca of the first earthworm, and these sperms, they remain stored, and when the eggs, they get mature, they get fertilized by the sperms of another individual. So why they cannot self-fertilize? One of the reason is that the sperms, they get early mature as compared to eggs during the breeding season. So it means the sperms are mature, but they cannot fertilize eggs. So they are transferred to between individuals, and later on when egg get mature from spermatheca, these sperms, they could come out and fertilize eggs. Another prominent structure that could that we can see here on the clitellum, on the ventral side of the clitellum, it is the tubercula pubertatus. So it is with related to puberty. So the animals that get mature, that is the earthworm, their clitellum at the ventral side would have glandular structure swelling, and these swellings, they would release the secretions So it is only present in the breeding season, it become prominent. So it is also showing the maturity of the, that is tubercula pubertatus. It is showing the maturity of the earthworm. On the earthworm, this tubercula is present, we would say it is a mature earthworm. It is sexually active earthworm. We are present in earthworm for locomotory function, but there are the genital setae present on the ventral side, and they are a little bit up toward from clitellum, and they are modified type of setae. They are broader than the normal setae. They are thickened, they are curved and hook like bristles. They help to uh, keep the body, the two worms, anchored and aligned while copulating. So here we can see these genital setae. So there are two pairs on this earthworm at the 25th segment at the lumbricus terrestris. So it is upward toward interior side from clitellum. So these setae, they are not locomotory. They would only appear during the breeding season in sexually mature earthworms. The two earthworm, they align ventrally, and the head and the tail of the two earthworm are in opposite direction. And the mucus from the clitellum of the both earthworm, it keeps them bind. So mucus is a sticky glycoprotein material. It is released from these tubercula pubertatus, that are the swelling of the at the ventral side of the clitellum, and they secrete mucus. So these mucus sheets, they align these, they keep them bound together, and ventral sides of the worms are attached to each other. So genital setae near the uh, clitellum, it anchors the two worms. So the genital setae of one earthworm, they would penetrate into the body of another earthworm, and the vice versa. So the earthworm, they are now bound with genital setae, and they are bound with the uh, mucus. So this firm grip, it will allow proper exchange of sperms from the male genital pores, and we know the sperms, that are produced in one worm are stored in its seminal vesicle, and they would come out from the genital opening of one worm. For example, from this worm toward anterior side on the segment number 15, it is the genital pore, that is male genital pore. So the sperm, they would come out, they would run along the seminal grooves, and they would reach to clitellum, and from clitellum they would get the mucus, and they would enter into the uh, semina spermatheca of the other individual. And similarly, the sperms from genital pore of the first earthworm, they would come out, they would run along the, from the male genital pore, they would run along the length of the body along the seminal grooves, and reach to the clitellum and enter into the spermatheca of the second individual, and they would remain stored. These animal, they would get separate, and then the sperm that are gathered from the other individual, when their own eggs get mature, these sperms, they come out and they fertilize their eggs, and these eggs, then they would make the fertilized eggs, they run in the seminal grooves and reach to the uh, clitellum, where they get mucus, and they make a cocoon of mucus around them, and they are shed in the environment, and later on they get hatched. The morphological features, we have to dissect the earthworm, and for dissection we have to use the wax bottom dish and using small pins for attachment, and a magnifying glass for observing the internal structures. We could re-anesthetize the earthworm in 10% ethanol if it is required. Then turn the worm dorsal side up. We have to cut because we cut invertebrates from dorsal side. They don't have any vertebral column. So we have to hold it in one hand, and we have to make a small slit using a scissor in the body wall in the mid dorsal line in the region of the clitellum. So we have to first identify clitellum, and then we have to cut it on the upper side of the clitellum toward head side and make a slit. And we have to be careful because it is very delicate organism, so scissor could go deep. We have to prevent the internal cutting. Keep the points of the scissors up, and we have to cut forward from clitellum toward the head up to the prostomium, that is a lobe like structure before mouth. And then we have to place this earthworm in dissecting plate and ventral side down, and we have to pin the skin on the sides of the earthworm to observe the internal organs. The cut earthworm in the dissecting plate, we can see internal organs. They are seminal vesicle and the spermatheca. They are visible, these white structures. Interior side, we start placing the pins on both sides in opposite to each other. So we have to spread the skin, and by using needle we can tease these uh, fibers that are attaching skin to the internal septa. So we have to remove these through pin, so that the skin could get clear from the internal organs, and then place the pins. So pin placing is to keep the body wall apart, and the angle of the pin should be oblique, so that while observing the internal organ these pins should not come in your way. And then we have to separate each septum from the alimentary canal using a teasing needle, and pin down each loosened bit of skin throughout the individual. First structure that appears, uh, and we probably see the first is the three pairs of cream colored seminal vesicle. Here we can see them, and they are used for storing the sperm produced by the same individual. This individual produce sperms, and they are stored in these seminal vesicles. So we have to tweeze them with the tweezer, and we have to remove them, and under that we could see the parts of digestive system. We should have the distilled water or the isotonic saline solution that should be poured on the whole individual, and the specimen should be submerged in water in order to avoid desiccation, because the desiccated organs they would not be easily visible. They would lose their structure and they would regress and not be visible. So we have to keep it submerged under water. We have to fill the tray with water. The earthworm, they are largely herbivorous annelid. They obtain food by eating in the soil uh, in its way and extracting nutrients from the soil as it passes through their digestive canal, and they take mixture of soil and organic matter through its mouth. So we have to first identify the mouth of the uh, earthworm. So here we can see the buccal cavity, and then after that is a structure that is muscular pharynx. So this muscular pharynx, it help in sucking action. It is attached to the muscles. So these muscles, they were torn by dissection, and the pharynx, it could look hairy due to those broken muscles. The esophagus, it is cylinder and it leads from the pharynx to the uh, thin walled broad swelling that is called as crop, that is for the temporary storage of the food material. The esophagus, it was hidden under the uh, reproductive organs and the pseudo hearts. So here we can see the pseudo hearts. These are the blood vessel like hearts. They are passing from one side of the esophagus to another side. We'll see them later. And then there could be certain calcareous glands present over here. They are called as calciferous gland or Moran's gland on the either side of the esophagus. So these glands, they could be in three pairs. Here they are not visible. So these glands, they help to remove excess calcium and carbonate from the blood that comes from the soil that is ingested by the earthworm. The crop is followed by muscular gizzard, and if we press the crop and gizzard gently, we can observe their firmness. The crop is soft and thin, the gizzard is muscular. So it help to grind soil and help in churning and the mixing processes. The gizzard, it would lead to the intestine, and this intestine may be called as midgut, and it is straight and thin, uh, that help in digestion and absorption. We can see the intestine, it is yellow in color because yellowish brown chlorogage cells, they are present in the on the intestine, and they give the intestine yellow color. And dorsal blood vessel could be uh, observable on the dorsal side of the intestine. These chlorogauge cells that are yellow in color, they work like the vertebrate liver, and they store glycogen and lipids, and uh, they are like the their function is like liver. And then the undigested material, it would go out from the posterior side of the intestine where the anus is present. More clear and magnified version of the previous slide. So all the organs are visible. So we can see here the pharynx and the buccal cavity, and the and here are the spermatheca and seminal vesicle, three pairs, interior, middle and posterior seminal vesicle are observable, and then there are anterior spermatheca and posterior spermatheca visible, and we can even see the pseudo heart that are present on the dorsal side of the esophagus, and uh, then we can see the uh, calciferous glands present here. So they are to clear the calcium and carbonate. Then here we can observe the crop and the gizzard. Crop is delicate, gizzard is firm, and dorsal blood vessel is observable on intestine and on the gizzard and the crop. So here we can see chlorogauge cells that act as liver. They store glycogen and lipids. Intestine here is visible. Better version of the previous slides. Here we can see the buccal cavity, and then the subpharyngeal ganglion. They are visible. The nervous system, circumpharyngeal connectives, they are also visible, that are connecting the subpharyngeal ganglion and suprapharyngeal ganglion. And then there are visible prostomal nerves. Pharynx is visible, and the body wall or septa, it is visible, and pseudo hearts, we can see them, and the segmental nerves, segmental ganglia, they are present. Anterior spermatheca, posterior spermatheca, where the sperms of the other individual would get stored while copulation. And then seminal vesicles, they are very prominent. You can see anterior, there are three pair, uh, that is anterior pair, middle pair and posterior pair of seminal vesicle. And we can also see the calciferous glands between these organs. The earthworms, as we know, they are monoecious. They have both male and female reproductive organs. We have to see first the male reproductive organs. In male reproductive organ, three pairs of seminal vesicles are easily observable, and they are located at the segment number 9, 11 and 12. And these seminal vesicles, they store and mature the sperms before copulation, and they are lying close to the esophagus. We can easily locate them. And then there are two pairs of testes that are present on the ventral part of the seminal vesicles in the sperm reservoir. We can see them. And sperm funnels, they collect sperms that are mature spermatozoa from the seminal vesicles, and then they transfer them to the efferent ducts. They are transferred into the two small vas deferens or sperm ducts, and these sperm ducts, they contain they carry the uh, sperms towards genital pores in segment number 15. These structures are very small, they cannot be easily found. So sperm funnels could be found if the dorsal wall of the seminal vesicles are removed. The female organs are smaller than the male organs even. There are two pair of round, glistening white seminal receptacles or spermatheca. So these spermatheca, here we can see an anterior spermatheca, and then are the pair of posterior spermatheca. They are easily visible in segment number 9 and 10. They store spermatozoa of another individual after copulation. The ovaries, they are present in segment number 13, but we cannot find them because it is very hard to find them. and pair of oviduct with ciliated funnel that carry egg to the female genital pore in the next segment, that is segment number 14, will also not be observable. So during mating, sperm from one worm travel along the seminal grooves to the spermatheca of another worm. Fertilization of the eggs takes place outside the body in the cocoon as it moves forward over the body, picking up the eggs of one worm and its mating partner's sperm from the spermatheca. That is all about the dissection of the earthworm. The system of the earthworm is concerned, they are having closed circulatory system. We have to locate the uh, pseudo hearts. We have to uh, they are also called as the aortic arches over the esophagus, and they appear to be uh, red because they contain the blood. They are pumping organs of circulatory system. We have to tease away the tissues to expose the arches. uh, that is, the even reproductive organs, they are located on the uh, these pseudo hearts. So we have to see these 5 pseudo heart, and then we have to find the dorsal blood vessel. Dorsal blood vessel is, we saw it is running on the dorsal side of the intestine. There would be present a ventral blood vessel, but we cannot see it because the digestive system, it has covered it. We have to retract the digestive system toward one side and see the ventral blood vessel underneath it. We will see it in next slide. The dorsal vessel, it is dark brownish red color blood vessel that is running on the dorsal side of the intestine. It could be easily observed, and then there are smaller blood vessels that are exiting from the dorsal blood vessel. But we cannot see ventral blood vessel from dorsal side at the intestine and the digestive system. It is covering this ventral blood vessel. So we have to retract the uh, intestine on one side, and we can see the impression of the ventral blood vessel on the ventral side of the intestine, and we can also see here the ventral blood vessel clearly. And when we retract the nerve cord, we can see the subneural blood vessel, and then there are latroneural blood vessel appear that is running along both sides of the nerve cord. So dorsal blood vessel would act as the vein, and the ventral nerve blood vessel, it acts as an artery, or it is like aorta. The blood from the aortic arches or the heart would enter into the ventral blood vessel, and from where the blood would enter into the capillary bed. Here we can see the capillary bed, and circulate in the body, and then come into the dorsal blood vessel and re-enter the pseudo heart. So dorsal blood vessel, it brings blood back towards pseudo hearts, and ventral blood vessel, it takes blood away from the pseudo hearts. We cannot see any respiratory organs, no gills or lungs, as they are not present. So the earthworm, they are not having any respiratory system. Gases are exchanged between the circulatory system and the environment through the moist skin. The excretory system of the earthworm are the tiny tubular nephridia. They are also called as metanephridia because they have both opening, that one is nephrostome and another is nephridiopore. Nephrostome is a funnel like structure that opens in one segment through septa, septum, and then the rest of the tubule is present inside the rear segment, and then there is a nephridiopore, that is a pore that is opening outside of the body. So each segment has two pairs and of the nephridia. These funnel-shaped nephrostomes, they have cilia to attract the coelomic fluid that is present here, and then coelomic fluid enter into the nephrostome, flows through the narrow tubes, and the blood vessels that are capillary bed is running along this structure, and then the fluid they go, they are the material that are useful are reabsorbed, and rest of the excretory waste they would go out from the nephridiopore. And here near nephridiopore the tubes are bulged and they are making a collection bladder, and then it opens it to the nephridiopore. These nephridia may appear as the yellowish color tubes, and they are very tiny, so they are not clearly visible. We have to see them carefully. For observing the nervous system, we have to first find the white suprapharyngeal or cerebral ganglia. We can see here they are called as pharyngeal because they are near to the pharynx, and they are uh, partially hidden by the muscles, so we have to remove those muscles to see them. But if we cannot see, it means we have destroyed them while cutting the earthworm. And then we have to observe the wide prostomial nerve, nerves that are extending from the cerebral ganglia, and a pair of circumpharyngeal connectives and extending from the cerebral ganglia under the pharynx. Then we have to locate the ventral nerve cord by pushing aside digestive system. Now we have removed the digestive system and pushed it away from the ventral side. We can easily see this thread-like nerve cord, and it is running throughout the length of the earthworm. We can observe the subpharyngeal ganglia, and then we can see segmental nerves from the ganglia that are present in each segment. Each segment has a slight uh, enlargement that is also called as a ganglion, and these ganglion they are extending lateral segmental nerves.